students, I am going to talk you through this experimental, the experimental variables page for today. Okay? And then, so this is your assignment for Monday. It's just independent learning. And it says experimental variables. What is measured during a controlled experiment? Why? When scientists set out to do an experiment, they first think about the variables that may affect the outcome of the experiment. A variable is any condition that may cause a change in the system being studied. Some variables are measured quantitatively, like temperature, mass, or height. So a note, quantitatively means with numbers. Okay, quantitatively means with numbers. The most important factor is that a scientist, other variables are recorded in a qualitative manner. Okay, qualitative. It means with words or descriptions. Okay. The most important factor is that the scientist runs a controlled experiment. In a controlled experiment, only one variable is changed to ensure that the effect of only that one variable can be measured. Okay, so a variable, the variable is the thing that can be changed. Okay, and then we measure the change either quantitatively with numbers or qualitatively with words and descriptions. A controlled experiment is when only one variable is changed. Okay, we've been talking a lot about that the last week, where we want one variable, we can only change one thing. This says Model 1, photosynthesis in an aquatic plant. Remember, aquatic means water, so a water plant like kelp. This is oxygen gas. That's the little bubble here. These little bubbles are oxygen gas, and this is a light. Number 1, the diagram in Model 1 illustrates a clipping of an aquatic plant in water. A, what process is occurring in the plant cells to produce the gas in the bubbles that appear? So what's happening? What's happening here? B, what gas is the plant producing? C, what source of energy is the plant using to conduct the process recorded in Part A? So where is the plant getting its energy from? And remember, plants get their energy from light. Question 2, depending on the environment the plant is in, more or less gas may be produced. Suggest a method for measuring the rate of gas production from the aquatic plant in Model 1. So how could we measure the gas produced by this plant? What could we do to measure the gas? Number 3 says, with your group, create a list of environmental factors that may affect the rate of gas production in the aquatic plant in Model 1. These factors could become variables in an experiment. So in here, we're like, so what could happen? What are the different things that could be changed? The amount of water could be changed. The amount of plant can be changed. The type of light can be changed. The amount of light can be changed. So what else can be changed? What could be a variable? In Model 2, aquatic plant experiment. So I'm looking at my pictures and I'm looking to see what's different in all of the pictures. I know that this is my aquatic plant, my kelp. I know that this is water. I know that the little bubbles here, is the bubbles represent oxygen. Okay, and here's my lamp, my light. Examine the 4 trials shown in Model 2. Identify several conditions in the experiment that are the same in each trial. Now I'm going to look at the picture and see what hasn't changed in the picture, okay? Describe the one condition that has been varied among the 4 trials. So what's the one thing here that's changed? Now the type of light hasn't changed because all 4 experiments have the same light. So that's not what has changed. Number 6 says, how does the condition described in question 5 appear to affect the rate of gas production by the aquatic plant? Provide specific evidence from Model 2 to support your answer. When I look back for my evidence to answer question 6, I'm going to be looking at the bubbles, okay? Because the bubbles are the oxygen being produced. Right? Was that oxygen? Yeah, the oxygen gas. Sorry about that, guys. And what do we notice about these? The amount of oxygen being produced in each part of the experiment. So you're going to provide specific evidence. You're going to use precise language to answer number 6. So refer to the diagrams in Model 2 to complete the data table in Model 3. Well, let's read the headings of our data table. Model 3, aquatic plant data. We have length of clipping, all the same, 12 centimeters. So that didn't change. Number of leaves on clipping, lamp power watts. Look, the lamp power watts are all the same, 40, 40, 40, 40. Percentage of light from lamp that reaches the plant. In A, we have 100%. B is 75%. C is 50%. And D is 25%. The number of oxygen bubbles formed in 10 minutes. So you're going to look up here to answer these questions. Remember, this is A, B, C, D. Okay. The column headings in Model 3 each describe a variable in the experiment. Okay, these are the headings. What variable was purposefully changed in the experiment? So which of these 5 variables was changed? What variable changed as a result of changing the variable listed in Part A? So what else changed? One of these variables was changed in the experiment, and that caused a change in another variable. What variables in Model 3 data table remained constant among all the trials? So constant means not changed, means the same. So what variables in the Model 3 data table remained the same among all the trials? Read this. When designing an experiment, you need to consider 3 types of variables. The independent variable is changed by the experimenter in the design of the experiment. This variable is sometimes called the manipulated variable. The dependent variable is what changes as a result of the change in the independent variable. This variable is sometimes called the responding variable. In some cases, more than one dependent variable is considered. The third category of variables is controlled variables. These variables that these are variables that you think may change the outcome of the experiment, but since they are not being studied, they need to be kept constant in each trial. Number 9. So let me just recap here. Let me go to the text box. Okay. So the independent variable variable, the independent variable. This is what is changed. Okay? The variable, the dependent variable, what happens because of the change. It's what is being observed. Okay? Controlled variable are what is kept the same in the okay? Number 9. Identify the independent, dependent, and controlled variables for the experiment that produced the data in Model 3. Independent, dependent, controlled. If you're stuck here, you have answered these questions in your previous work, okay? What you're doing now is just defining them a little bit differently. You're using these words, independent, dependent, controlled, to define. Read this. A well-written research question states the independent and dependent variables in the experiment. For example, a student investigated the effect of soil pH on the number of strawberries produced by a strawberry plant. Her research question was, how does the pH of soil affect the number of strawberries produced by a strawberry plant? Write the research question using the format suggested in the Read This box for the experiment in Model 2. So again, we're going back to the experiment in Model 2. And we're going to use the if then because model. Okay, we use that in class, especially when we did the tomato juice on the head. Okay, so write a research question for the plant experiment in Model 2 using the if, then, because format. Number 11 says, a student wonders, does the moisture content in soil affect how far a worm can dig? Identify the variables that are being considered in this experiment and the variables that need to be controlled. So again, the independent variables are what you're going to keep, are what you're going to change, manipulate it, okay? Dependent variables are what is what you're observing, what you're looking for, and controlled are the things you're going to keep the same. There you go. Now I'm going to show you how to save this to your Google Drive. I'm going to click Save, Add to my drive. Okay, now the name is up here. All right, I'm going to add my last name just so I know, then Add to my drive. Okay, it wants to know where I want to put this. There's a folder called Kami Assignments or Kami Uploads. You have one called Kami Uploads. It says Kami. Or put it wherever you want, somewhere you will know. Okay, I like the Kami Uploads because I remember it's got all my PDFs in it. Then I hit select, it goes there. Then when I go to Google Classroom to share it, I have to find it in my drive. But it should be pretty easy to find if you use recents, because it will be the most recent item in your Google Drive. I hope that made sense. So I hope that this helps break down this assignment for you. I know this is an independent learning activity on a Monday, but if you need me, just shoot me a message through Google Hangouts or an email, and I will happily answer your question as soon as I get it. I hope you all had a great weekend, and I will see you on Tuesday in biology. Now let me figure out how to stop recording. This is always the challenge, right? I've said that like 10 times to you guys. By now you'd think I'd know.